Welcome to The Explainer. Today, we're going on a totally wild phylogenetic safari through a world that is bizarre, alien, and yet literally right beneath our feet. Take a look at the blue dragon. It's a type of sea slug with these striking colors and thin, finger-like structures that help it just float upside down on the ocean's surface. But here's the absolute kicker. It hunts the highly venomous Portuguese man-of-war, absorbs its stinging cells at a molecular level, and then uses those exact same cells to deliver a deadly sting of its own. I mean, it's surreal, right? And the most amazing part? The blue dragon doesn't even have a backbone. It's an invertebrate. Okay, let's dive into this. Our planet is defined by a pretty staggering reality. Animals without a backbone account for over 95% of all known animal species on Earth. We humans, along with every bird, fish, reptile, and mammal you can possibly think of, well, we're crammed into that tiny little 5% sliver. It is very much a spineless world, and we're just living in it. So here's the roadmap for our safari today. We'll be covering rulers of the animal kingdom. 2. Sponges and ancient jellies. 3. The diverse Lophotrochozoans. 4. Arthropods and the molting masses. And finally, 5. Our surprising sea star cousins. Let's get right into it. Section 1. Rulers of the animal kingdom. To really grasp the sheer scope of these creatures, we have to recognize that they aren't just numerous, they are literally everywhere. As this quote highlights, invertebrates occupy almost every habitat on Earth. We're talking everything from the scalding water released by deep-sea black smoke or hydrothermal vents, all the way to the completely frozen ground of Antarctica. Evolution in these extreme environments has produced an immense diversity of forms, ranging from microscopic life forms to massive giant squids spanning 18 meters long. They absolutely rule the planet. Section 2. Sponges and ancient jellies. Let's start right at the base of the animal tree, with the ancient life forms that laid down the blueprints for everything that followed. First up, the phylum Porifera, commonly known as sponges. Sponges are incredibly simple. In fact, they're so simple they actually lack true tissues altogether. Instead, they rely on specialized cells called choanocytes, which use flagella to sort of draw water through their bodies, filtering out food particles. But, don't let their simplicity fool you. They have these amazing totipotent cells called amoebocytes that can basically turn into any other kind of sponge cell, giving them this incredible biological flexibility. And on a molecular level, sponges are absolute chemical powerhouses. They synthesize complex defensive molecules like cribstotin. Molecular studies have actually shown this can kill cancer cells and even penicillin-resistant bacteria. Stepping one rung up the evolutionary ladder, we encountered the conidians. These guys originated about 680 million years ago. Now, unlike sponges, these animals do have true tissues, which are organized in a diploblastic radial body plan. They generally exist in two main variations. You've got the stationary wait-for-prey polyps, like sea anemones, or the free-swimming medusa, like jellies. This highlights a huge evolutionary leap. They developed a gastrovascular cavity for digestion and an unbelievable molecular weapon called cnidocytes. These are specialized cells containing nematocysts, which essentially act like microscopic explosive harpoons that inject poison right into their prey. Section 3. The diverse Lophotrochozoans. We're now entering the realm of the bilaterians, animals with bilateral symmetry. Okay, Lophotrochozoan is definitely a bit of a mouthful, right? But the crucial point to remember here is that this massive group wasn't clumped together just because they look alike. Actually, they are the most physically diverse clade of animals out there. The Lophotrochozoan clade was identified through deep DNA and molecular data analyses. Sure, some members share physical traits, like a ciliated feeding crown known as a lophophore or a unique swimming larval stage called a trochophore, but really, it is their genetic molecular footprint that links 18 wildly different phyla together. Just look at the extreme diversity within this one single clade. First up, we have platyhelminthes, or flatworms. Because they're completely flat, they don't even need a circulatory system. Gas exchange happens via direct cellular diffusion. And some of these, like parasitic blood flukes, use molecular camouflage. They actually mimic the surface proteins of their hosts to evade human immune systems, letting them survive inside us for up to 40 years. No way, right? Then we have Annelida, segmented worms with closed circulatory systems. This includes leeches that produce a highly powerful molecular anticoagulant called hirudin. Finally, mollusca. They share a basic body plan, a muscular foot, visceral mass, and a mantle, but they've evolved into everything from your everyday garden snail to massive, highly intelligent giant squids that battle sperm whales in the deep ocean. Section 4. Arthropods and the molting masses. Let's move to our next major clade, the ecdysozoans.
This group contains the most species-rich animal phyla on the planet. The defining feature of this clade is a process called ecdysis. Because these animals have a tough external coat, an exoskeleton, they can't just smoothly grow larger. They have to undergo this highly energetically expensive process of shedding their old coat to grow a new one. This group includes nematodes, or roundworms. And get this, some parasitic nematodes have an extraordinary molecular toolkit. They can literally inject molecules into their host's muscle cells to regulate gene expression. This makes the host cells highly elastic and actually forces the host's body to grow new blood vessels just to feed the worm. It's biological hacking at its finest.
But the true kings of the ecdysozoans are, without a doubt, the arthropods. I want you to let this number sink in for a second. 1 quintillion. A billion billion. That is the estimated number of individual arthropods currently living on Earth right now. Insects, spiders, crustaceans. Their success is largely driven by their segmented bodies and hard exoskeletons. And what's absolutely fascinating at the molecular level is that their massive diversity didn't come from acquiring brand new Hox genes for body segmentation. Researchers found that arthropods and their ancestors actually shared the exact same Hox genes, like UBX and AB-A. It was changes in the sequence and the regulation of those existing genes that drove the incredible diversity of arthropod body segments we see all around us today. Arthropods, specifically insects, also mastered the air and the land through some pretty incredible developmental strategies. In incomplete metamorphosis, the young look like tiny adults and just molt until they get wings. But in complete metamorphosis, the larval stage, like a caterpillar, looks entirely different from the adult butterfly. During the pupal stage, the larval tissues are entirely broken down at a cellular level and completely rebuilt. This strategy meant the larvae and the adults didn't have to compete for the same food sources, which allowed insects to just absolutely radiate and conquer the planet alongside flowering plants. Section 5. Our surprising sea star cousins. For our final stop, we move to a branch of the phylogenetic tree called Deuterostomia. Now prepare to be a little surprised about who we share this branch with. Deuterostomia is a clade defined primarily by molecular and DNA similarities. And, incredibly, that DNA tells us that echinoderms, sea stars, sea urchins, and sand dollars are grouped with chordates. And chordates include us vertebrates. Echinoderms are exclusively marine. They have a thin epidermis covering a hard calcareous endoskeleton. But their most unique feature has to be their water vascular system. This is a network of hydraulic canals that branch out into hundreds of tiny tube feet, allowing them to move and grip with tremendous force. And while they look radially symmetrical as adults, their larvae are actually bilaterally symmetrical, which is a big hint at our shared evolutionary past. Let's see how they put that water vascular system to use. It involves a visceral, almost alien mechanic. Because how does a sea star eat a clam that's much bigger than its own mouth? Well, step 1, it uses those hydraulic tube feet to firmly grip the closed shell. Step 2, it literally turns its stomach inside out, averting it out of its mouth and slipping it right into the narrow crack of the bivalve's shell. Step 3, it secretes digestive juices to liquefy the mollusk alive inside its own shell, and then just pulls its stomach back in. It's a terrifyingly efficient adaptation. However, we do have to end our safari on a somewhat more sobering note. Despite their vast numbers and incredible molecular and physical adaptations, invertebrates are highly vulnerable. When we look at documented human-caused animal extinctions, we obviously hear a lot about endangered mammals and birds. But mollusks alone account for a staggering 40% of all documented animal extinctions. Freshwater bivalves and terrestrial gastropods are being increasingly wiped out by habitat loss, pollution, and overharvesting. Which leaves us with a pretty profound final question to ponder. Insects, mollusks, sponges, and arthropods, they own the Earth. They are the hidden 95% majority that make our ecosystems actually function. But how much of this spineless world is going to survive our impact? It's a stark reminder of the incredible invertebrate foundation that all of life is built upon. Thank you for joining me on this explainer, and keep your curiosity alive.